Today's lecture will be on the structure and function of the digestive system. So the function of the digestive system is first the ingestion, usually occurring in the oral cavity. It's the first process of digestion and absorption. Then we have digestion itself. It's mechanical digestion where we see mastification occurring in the oral cavity. Chemical digestion with the mixing of enzymes from saliva. Another function would be propulsion, which we see, which carries the food, the bolus of food down through the esophagus, eventually reaching the stomach where we begin the next function of the digestive system, which is secretion of digestive enzymes into the small intestine, where we start the next function of absorption. Absorption is basically passive or active transport of digestive products across the GI tract epithelial tissue, and then eventually making its way through the large intestine for the last function, which is going to be elimination of waste through the feces and defecation. So when we're looking at the different types of digestive organs, the first one we'll look at is going to be basically the digestive organs themselves. And the main aspect with the digestive organs is that they're all housed within the gastrointestinal tract, or they collectively make up the gastrointestinal tract. We see the oral cavity as part of it, the pharynx, the esophagus, the stomach, large intestine, small intestine, and eventually out through the anal canal. The accessory digestive organs are not part of the long GI or gastrointestinal tube, but often develop as outgrowths of that tube. So what we see, these are accessory, so the teeth with mastification, the parotid salivary glands, the tongue, sublingual and submandibular salivary, the liver, the gallbladder, and the pancreas. So each one of these are accessory to aid in the digestion and propulsion and secretions of the digestive material into the GI tract. So when we look at the difference between peristalsis and mixing, so when we look at peristalsis, we're looking at a ripple wave of muscular contraction, remembering that the smooth muscle of the GI tract is what is going to change the overall lumen size, or the size of the lumen. You can picture this like a toothpaste. It's basically essentially contracting, pushing the bolus of food forward, and then we see a relaxation, and then another contraction and pushing the bolus of food forward. Basically, its job to move the bolus of food further through the GI tract. When we get into mixing, also known as segmentation, is a combination of peristalsis, but then we also have the churning of materials. And the churning of materials does two things. It slows down the rate of the bolus or the chyme moving through the digestive, so allows for absorption. And it also helps mix the secretions that it picked up through the stomach and the small intestines. So we usually see these digestive secretions occurring most often in the stomach and in the small intestine. So the first line, or what we consider to be the first line of digestion, is going to occur in the oral cavity. And the reason we consider this to be the first line of digestion in the oral cavity is because in the oral cavity what we see is a mechanical and chemical breakdown of food. So the first part that we're going to talk about are the inferior and the superior lip. These are made of keratinized, stratified squamous epithelial tissue. Remember, keratinized means that eventually on the surface of the lips we're going to see that you lose skin, such as chapped lips. And then we have the gingivae, which are located or otherwise known as your gums. The gingivae are more of a dense, irregular connective tissue. And the lips and the gingivae are connected by the labial frenulum, this little part right here. And when we're looking at the labial frenulum, we see the connection between the gingivae and the lip. And we see that in both the superior and the inferior side. The next part we'll talk about is going to be the palate. So you have two palates. The first part, the anterior 2/3 of the upper roof of the mouth, is going to be bone. We consider that to be the hard palate. And then the posterior 1/3 is going to be more muscular, and this is what we call the soft palate. Another important part are these transverse palatine folds. They're located in the upper roof of the mouth. They're important. They assist the tongue, so it helps with manipulating food when it's in the mouth. So it creates a breaker. So as the tongue is manipulating food, the transverse palatine folds help hold the food inside of the oral cavity. The uvula, I'll point it out here. This here, or when you're looking at from an anterior view of the uvula here. The uvula works in combination with the soft palate. So we see that the soft palate and the uvula elevate whenever we swallow, and it's basically going to elevate and close off the nasopharynx. So what we see is that when you swallow, the soft palate and the uvula will help close off the nasopharynx, and it'll allow the food to go through the oropharynx, eventually into the laryngopharynx and into the esophagus. So when we're looking at the different types of salivary glands, we consider these salivary glands, the parotid salivary, the sublingual salivary, and the submandibular salivary glands, to be extrinsic glands, meaning that they're located in the oral cavity, but they're outside or on the extrinsic surface of the oral cavity. The first one is the parotid salivary glands. It's located anteriorly and inferior to the ear. It secretes about 25 to 30% of our total saliva. And you can see here how we also have a duct that runs parallel to the zygomatic arch, and eventually it's going to pierce the buccinator muscle, and then it's going to empty out just secondary into the roof of the mouth, just secondary to the upper molar. It secretes an enzyme called amylase, a digestive enzyme that helps stimulate the stomach for digestion. The submandibular is our next biggest one, or is the biggest one. It's going to produce the majority of our saliva. So the submandibular salivary gland is going to produce about 60 to 70% of our saliva. It lies inferior to the body of the mandible. It also has a submandibular duct. So when we're looking at the submandibular duct here, and this submandibular duct transports the saliva from each of the gland through the papilla in the floor of the mouth, and it's lateral to the lingual frenulum, and the lingual frenulum is what attaches the lip to the gingivae. So it's just right in front of your lower lip. And then finally we have the sublingual gland. I'll erase this here. And so we have the sublingual gland, and then we also have these sublingual ducts. And some characteristics of the sublingual gland are that it's just inferior to the tongue. These ducts that open onto the inferior surface of the oral cavity, just posterior to the submandibular duct. And so we see the submandibular duct empty out more in the anterior portion, and we see the sublingual salivary ducts emptying out more inferior. And the sublingual salivary gland only contributes about 3 to 5% of our total saliva. So when we're looking at salivary gland secretions, there are two types of secretions or cells that are found in salivary glands. When we're looking at the parotid glands are going to be more of the serous. And when we're talking about the submandibular or the sublingual glands, these are going to be mostly the mixed glands. And what that means is that they're going to produce both the mucus and the serous fluid. So first, when we're talking about the serous cells, they secrete a watery fluid. They contain ions, lysozymes, and salivary amylase, basically digestive enzymes. Again, the parotid glands are most often associated with the serous cells. When we look at mucus or mucose cells, they secrete mucins, which becomes mucus once it mixes with water, which forms mucus upon hydrating with water. So when we look at these two, we can see that we have a row of serous and then a row of mucose, and then these are going to be considered a mix. We usually find these in the submandibular and the sublingual. So when we're looking at the walls of the GI tract, mostly what we're looking at is the GI tract from the esophagus to the large intestine, and they're composed mostly of 4 concentric layers called tunics. So we have the mucosa, the submucosa, muscularis, and the serosa, or sometimes the adventitia is another name for it. And we're going to start first with the mucosa. It's the deepest one of them. With this, what we see in the mucosa, it's the deepest layer. It has a layer of superficial epithelium. With the superficial epithelium, it also has an underlying layer of the lamina propria, and the lamina propria is composed of areolar connective tissue. And then we have a thin layer underneath called the muscularis mucosae. And with the submucosa, we're just working our way out. We're going to have components of the lymphatic ducts. So this is where the MALT, the mucosa-associated ducts, are located, or the Peyer's patches. So here we have the submucosa gland, which is a mucin secretion gland. And inside of these, we're also going to have the lymphatic ducts. So any of your fat-soluble material is going to enter into this, and then into the lymphatic system, and eventually into the lymphatic system, into the lymphatic vessels. We also have a high concentration of blood vessels, and then we also have a nerve plexus, and nerve plexus is important. It's going to be the first associated ganglion. We have both the sympathetic and parasympathetic system in the submucosa nerve plexus, and it's also called a Meissner plexus. And then working down, we're going from deep to superficial. We're going to have the muscularis. It's basically 2 layers of smooth muscle, and the first one is going to be the thickest. So we have here the inner layer, and the job of the inner layer, inner circular layer, is going to be to constrict the lumen and forms the sphincters. So it's actually what is going to change the overall diameter of the lumen. And then the outer longitudinal layer is going to shorten the tube. So when we talked about mixing or segmentation, you usually have two components. So the inner circular layer is going to shorten the tube, or shorten the diameter of the tube, and then the outer longitudinal layer will shorten the entire length of the tube. This process allows for the bolus of food to slow down, increasing the absorption rate through the submucosa layer. The only exceptions we see is the esophagus. So in the esophagus, it has not only smooth muscle, but some of skeletal muscle. And then the stomach contains all three layers of smooth muscle. So those are the only two exceptions when we're looking at the different tunics. It also has a nerve complex. So here, this myenteric nerve plexus, also called an Arbok plexus. This nerve plexus is what is going to activate both the inner and the outer smooth muscle layers. And then we have the serosa, and sometimes it's the adventitia. It's an areolar connective tissue made up of collagen and elastic fibers. So it allows for, when we see digestion occurring, for the elastic property of the small intestine, large intestine to be able to stretch. And so that was a short video on the structures and function of the digestive system.